Hi students, now in this class we have to talk about the very important component of the immune system that constitutes the third line of defense, what's called antibody. So the antibodies are chemically immunoglobulin proteins. They are immunoglobulin proteins synthesized and exposure to antigen. Then once formed, they combine specifically with the antigen. Whenever pathogens enter in the body, the B lymphocytes produce an army of proteins. So the nickname for antibody, army protein. Army protein. As it is fighting against the antigens. So the nickname for antibody, the army proteins. So whenever pathogens enter in the body, the B lymphocytes, they produce antibodies, what are called the army proteins, to fight against them. Okay, so the antibodies are secreted on exposure to antigen by the effect of B lymphocytes called plasma cells. I mentioned earlier, at the time of functioning, once the B lymphocytes are stimulated, they form two types of cells. One cell, the plasma cells, effector cells. Another cell, the memory cells. The plasma cells only produce antibodies. So they are secreted in response to antigen by the effect of B lymphocytes. What we can see, there is a plasma cells. Now let's see the structural aspects. So it was Thomson Porter and then Edelman in 1950s exposed or revealed the structure of that is immunoglobulin molecule. They explained the Y-shaped structure of immunoglobulin molecule, particularly that is what we call D, E, G. These are all the types. So monomer, they describe the monomer, the molecular structure of that is what is called that is immunoglobulin molecule, the Y-shaped molecule. So it is a glycoprotein, a protein one. If we are taking any protein, we can have polypeptide chain. In a polypeptide chain, one end we have N terminal having free amino group. Another end we have free carboxyl group, that is called C terminal. That's the nature of the polypeptide chain if we are taking. So we have N terminal having amino group, free amino group, C terminal having free carboxyl group according to their name. So here this Y-shaped molecule is formed of 4 polypeptide chains. 2 chains are called light chains. Now you see that one, this is the light chain. This is the light chain. Or you can see this is the light chain. L chain. 2 chains are called heavy chains. So these are all called the heavy chains or L chains. So each immunoglobulin molecule is formed of 4 polypeptide chains of 2 different types. A pair of that is light chains and a pair of heavy chains. Okay. Now each chain is different from one another in length as well as the number of amino acids and also the molecular weight. Sorry. So if we are taking L chain, it is having a molecular weight of 25,000 Dalton. This is the molecular weight. Having nearly about 214 amino acids, constant number of amino acids. The L chain is having constant number of amino acids with a molecular weight that is 25,000. If we are taking the heavy chain, the molecular weight is about double just what we have the light chain, that is 50,000 Dalton, with a varying number of amino acids from one antibody to another antibody. So an example of 420 to 440 amino acids. So in the book it is given that is 450. Okay, so the L chain is having a molecular weight of 25,000 Daltons with a constant number of amino acids 240. The heavy chain is having a molecular weight of 50,000 Dalton with varying number of amino acids from one antibody molecule to another antibody molecule, ranging from 420 to 440, but in your book it is given 450. Let's take that one. This is 1. Okay, so we are forming actually two polypeptide chains. Now this is the light chain I mentioned about. You see that one having only half of the length, and this is also the light chain. And you see that one, the long one is called as a heavy chain. So having a molecular weight, you see double that of what we call the light chain. Now the two chains are held together by means of disulfide bond. You see that one, the two heavy chains are joined with disulfide bond. This is a disulfide bond. So you see the disulfide bond is formed wherever you have the amino acids with what is called sulfur group. We have only two amino acids with the sulfur group. One is methionine, another one cysteine. Here you see that one, just the disulfide bond is formed wherever you have these amino acids, either cysteine or methionine is present. So the two heavy chains are joined together by means of disulfide bond. And likewise, it's also called as disulfide bridge. Likewise, one light chain is joined with one heavy chain, again by means of disulfide bond or a bridge. So disulfide bridge is present between the two heavy chains and also between one light chain and one heavy chain. The meaning for that one light chain is joined and attached to each of the two heavy chains. Now this is one light chain, one heavy chain. This is one light chain, this is one heavy chain. Okay, this is about the structural aspects. As we have two different types of what is called the chains. Now normally we are representing the immunoglobulin molecule Ige2L2. Ige2L2. The meaning for that one, the monomer. Monomer means a single molecule. The monomer is formed of actually two heavy chains and two light chains, Ige2L2. Now normally you see that one in a polypeptide chain, a peptide bond is present to form a long chain between the carboxyl group of one amino acid and the amino group of another amino acid. But at a particular region, the peptide bond is fairly flexible. As a result, the two heavy chains have a flexible region. That is called what is known as hinge. So we have a flexible hinge is present. Now this is what's called the hinge, where the peptide bond is flexible in each heavy chain, so that between the two heavy chains we have a flexible hinge at their approximate middles. So this is a flexible hinge that is formed because of the weak peptide bond in that what is called the heavy chain at that region. So this is about something about immunoglobulin. Now if we are taking immunoglobulin, we have two terminals, what I mentioned about regarding the polypeptide chain. One terminal where we have the amino group, a free amino group is called as the N terminal. So N-terminal, the polypeptide chain having a free amino group. So that is called N-terminal. At the other end, we have a free carboxyl group that is called as a C-terminal. C-terminal. So N-terminal, C-terminal regarding what we have the free amino group, free carboxyl group. Similarly, each molecule of immunoglobulin shows two distinct zones. One zone is called the variable region. What I just actually put a black color in both heavy and light chains. Here also I represented. So one region, so there are two regions. One region that is called normally what is called variable region. This is called as a variable region. So variable region. The meaning for that one, they show varying sequence of amino acids in different antibody molecules. Varying sequence of amino acid in different antibody molecules. Now another one, what we have a constant region, a large constant region. You see that one starting from here up to this one. So it is called FC, fragment of constant region. And this is called a FAB region, fragment of antigen binding site. Or we can say first FV region, variable region. I mentioned here the variable region FV. So fragment of variable region, the meaning for that one. They have actually varying sequence of amino acids. The number of amino acids is constant, but they have varying sequence of amino acids to differentiate different types of antibody molecules. And another one what we the long region, the large region, having what we call constant region or FC region, having what is called the same sequence of amino acids. This is with reference to two regions and the two terminals. So one terminal amino, another one carbox or C terminal. And we have two regions, one variable regions, both exhibited by, actually you see that one heavy and light chains. And also we give a large constant region with what is called constant number of amino acids. Variable region shows varying sequence only, not actually the number, varying sequence. They have same number but the sequence. Suppose we have for example methionine 1 and just what we have glycine 2, like that. In different what is called antibodies, we have varying sequence of amino acids, hence called variable region. But again, this is called as a constant region having the same sequence of amino acids. That's about the two regions. Okay, now I'll proceed further. On the basis of physiological and biochemical properties, the antibodies and immunoglobulins are classified into 5 different types. So, IgE, IgM, IgD, IgE, IgG. These are the 5 different types: immunoglobulin A, D, E, M and G. Now, these 5 types of immunoglobulins are biochemically and physiologically different, even in structures also. Now, Ig, D, E, G, these are the monomers. What they represent is nothing but a monomer, single molecule, a monomer, a single molecule. So, D, E, G are monomers. Then if you are taking just actually IgA, it is a dimer. It is formed of 2, that is immunoglobulin molecules. They are united by means of special proteins. Then the pentamer. If you are taking what is called IgM, it is a pentamer. Having, you see that one 5 different antibodies. They are all joined together by specific proteins. Now the first formed antibody during what is called infection is nothing but IgM. The first formed antibody, IgM. Okay. The most common type of antibody, that is IgG. The most common type of antibody, IgG. So it is the only antibody that can pass across placenta. The only antibody that can pass across placenta. That's why we have the problem during pregnancy. That is related to the orange factor, erythroblastosis fetalis. This is because of the movement of IgG antibody from mother's blood to the fetal blood. Then we have IgA antibody. This is formed in the colostrum. The first formed milk. The breast milk is the best milk for children, providing defense mechanism. This is because of the enrichment of IgA antibody present in the colostrum. It is also found in the mold, mucosa associated lymphoid tissue. It is rich in IgA. Now IgE, it is involved in what is called allergic reactions. It is involved in allergic reactions. And this is simple. So nature of the structure is different, monomer, dimer and pentamer, and some specific characters related to the entrance point of view. I'll write it once again on the board at the end. You can copy it down. Okay. So these are all the 5 different types. So this is one arm of monomer. This is another arm of monomer. So each arm of monomer, both in heavy and light chains. See, this is one arm. This is another arm. Each arm is formed by a heavy chain and a light chain. So each arm in a monomer, both in what is called heavy chains and light chains, combine together to form antigen binding site. This is the region during interaction between antigen and antibody where the antigen binds to what is called this immunoglobulin. I already mentioned that is paratome, the part of the antibody which makes contact with what is called the epitope, the part of the antigen. That is called FAB, fragment of antigen binding site. Now this is the antigen binding site. So the two, just actually what we have the light and heavy regions, the variable regions in each monomer. is having antigen binding site. I represent some cavities. So each antibody is having just specific shape to fit the antigenic determinant, to fit the specific antigenic determinant. Here it represented in the form of semicircle, in some cases in the form of square or rectangle, that is the cavities. The antigen binding sites are normally shaped, specifically shaped to fit for the antigenic determinant, that is epitope. Here I represented the structure of an antigen. So what is epitope? Epitope is nothing but the part of antigen, active part of antigen, antigenic determinant, which fits into the antigen binding site that stimulates what is called the interactions between antigen and antibody. So we have two antigen binding sites. Here one antigen binding site, here another antigen binding site. That is a site into which the epitope or antigenic determinant fits. So that is what is happening. So two antigen binding sites for antigenic determinants. The interaction occurs. Okay. So there are two antigen binding sites in a monomer, 1 and 2. Now what about this C region? The C region forms a long stem of what is called antibody molecule. The C region forms a long stem of the antibody molecule, which determine the antibody class. The antibody class means 5 types of antibody class. So they are differing. So in their number of amino acids. So this long stem, the constant region, determines the antibody class and have common characters and serve common functions for all antibodies. They serve common functions. But here the variable regions exhibit different functions because they have to fit for different antigenic determinants of antigens. That's about what we call the different aspects of what is called the antibody or immunoglobulin molecule. Here I added that is a part of the, so this part is a paratope, the part of the antibody. Now this region, what it represents is the epitope, antigenic determinant, that is a part of the antibody which makes a contact with the paratope, the part of the what is called antibody. So epitope is a part of antigen and paratope is a part of the antibody. So there is a binding between these two that stimulate the immune reactions. That's all about what we have the structural aspects of what is called the antibodies. On the basis of biochemical and physiological properties, there are 5 different types of antibodies. One, IgA alpha. It is a dimer, what I mentioned earlier, found in colostrum, the first formed milk, just the breast milk, that is pregnancy, after delivery, after parturition. It's also found in mouth, tears and saliva. The second one, delta, IgD. It is also a monomer placed in function. Then the third one, Ige epsilon. It is a monomer, it is involved in allergic reaction just like what we have asthma. And also involved in inflammatory responses. Then IgG, gamma, monomer, the most common type, the abundant type in the body. Only antibody that can pass across placenta. That is the reason why in the case of Rh factor, the development of erythroblastosis fetalis occurs. This is because of anti-Rh antibody. The anti-Rh antibody is of IgG type. Anti-Rh antibody is of IgG type that can pass across placenta and causes erythroblastosis fetalis. Then IgM, mu, it is a pentamer. It is the first formed antibody in the case of fetus as well as during infection. Later only they get assorted into what is called G type, the common type and other types. So it's a pentamer formed of what is called 5 immunoglobulin molecules. First formed antibody in fetus during development and also during infection in the body. That's about what we have. There is a types of antibodies based on physiology and biochemistry. antibodies or glycoproteins. Now what are the functions of antibodies? They affect neutralization of what is called the toxins of bacterial origin, agglutination, sticking together of the bacterial cells, precipitates cause the precipitation of soluble what is called the antigens, like what we have the toxins, anything else. Axonization, it is the meaning for coating over, modify the bacterium in such a way that they can be easily phagocytosed by the phagocytic cells. I want to discuss all these things the next part, what we have the interactions between antigen and antibody in detail.